Hello everyone. I'm delighted to be here to introduce our latest article published in the iMeta journal. The title of the paper is Soil Labile Organic Carbon Fractions Mediate Microbial Community Assembly Processes During Long-Term Vegetation Succession in a Semi-Arid Region. We know that microorganisms play a vital role in biogeochemical cycles, serving as a key link in sustaining above-ground and below-ground interactions within terrestrial ecosystems. They are also the game changers in the restoration of degraded soils. Microbial community assembly has been a focal point of research in recent years, as it significantly influences ecosystem functions. Community assembly is typically divided into deterministic and stochastic processes. Currently, the community assembly processes on long-term time scales remain unclear. Soil organic carbon is typically divided into two components, active organic carbon and stable organic carbon. Conceptualizing soil carbon components can better explain minor changes in management. Based on this, we propose scientific question. How do organic carbon components affect microbial community assembly during long-term vegetation succession processes, and propose hypotheses? We collected soil samples, a typical long-term successional sequence on the lowest plateau. These samples will be analyzed for soil bacterial and fungal diversity using amplicon sequencing. The research findings indicate that long-term vegetation succession significantly increased the accumulation of organic carbon components in the soil layer. The bacterial community is predominantly composed of proteobacteria, while the fungal community is dominated by Basidiomycota. NMDS analysis results indicate differences between various successional stages, with significant variations in the composition of both bacterial and fungal communities. The null model results suggest that homogeneous selection dominates the assembly of both bacterial and fungal communities, with deterministic processes in the surface microbiota being weaker than in the subsoil. The co-expression network results reveal that the surface co-occurrence network is more complex than that of the subsoil. Furthermore, our analysis of the microbial network identified a set of keystone microorganisms that play a crucial role in community assembly. The cross-domain microbial network results further demonstrate that long-term succession increases microbial network complexity. Simultaneously, the relative abundance of most key taxa shows a declining trend, and they exhibit a negative correlation with subcomponents and microbial assembly processes. Network analysis further indicates that active carbon components are more closely related to the microbial community compared to stable carbon components. Further, regression analysis results suggest that, relative to soft and stable carbon components, active carbon components are more effective in explaining changes in the microbial community. The PLSPM model results indicate that active carbon components are the primary driving factor for microbial community assembly, exerting this effect by influencing microbial characteristics. Key taxonomic groups of microorganisms have a significant impact on the assembly of both surface and subsoil microbial communities. Bacterial alpha diversity affects fungal community assembly. Based on the above results, the following conclusions can be drawn. Homogeneous selection governed microbial community assembly. The bacterial and fungal deterministic processes increased with soil depth. Long-term succession increased the complexity and stability of network. Labile organic carbon dragged the microbial assembly processes by affecting bacterial alpha diversity and keystone taxa. Welcome everyone to download and cite our paper. Thanks again for the iMeta journal.